Okay, I noticed we have kind of an accumulating amount of detritus up here that people leave. Probably, maybe after this lecture, maybe not. But if anything up here looks familiar, if you've lost something, don't be afraid to come grab it. There's a little bracelet here, whatever, pencil case. Okay. The next time we meet, we will be taking an exam. So if you can come a couple minutes early just to get settled. I like for students to put their backpacks off to the side, you know, grab an extra pencil or water bottle if you need it or whatever. We will be full, kind of, you know, everybody comes on test day, or we hope so. So we'll be a little more crowded. You should have plenty of time. The test has 56 multiple choice questions. It's a test that's designed to be given in 50 minutes, and I'm giving you the full hour and 15. So I think for the most part, you'll have plenty of time. But I have extra office hours tomorrow. I'll be around probably most of the morning, 9:30 to 11:30 at least. So come see me if you want. Office hours on Monday as well. Remember, your homework is also due Tuesday at class time. And I would recommend that you don't put that off till, you know, right before the exam. So get that out of the way in here the next, hopefully today, tomorrow, and then give your chance some time to, you know, be looking through the notes, looking through the learning objectives. There are lots of practice questions on the Mastering website. So you say, well, we did most of chapter 4. That was microscopy, taxonomy. You go into the study helps on chapter 4, and there's some practice questions, things like that. Now, of course, there are things in the book that we didn't cover in class that you might see practice questions on, and you could try to ignore that. I won't ask you on things in the textbook that we didn't cover in class. So, all right. I did want to just clarify a couple things. I had a few questions from last lecture that I thought were good questions that maybe other people wonder about. We went through, we talked about malaria, we talked about fungi, a lot of terminology, fungal forms, you know, filaments, aseptate, hyphae, all that. I was asked the difference between mycelium and mycorrhizae. Okay, so mycelium, remember, is a mass of filamentous hyphae. Oftentimes that's underground. So mushrooms are going to have a mass of mycelium underground. Molds, right? Molds are just made of mycelium, basically masses of hyphae. Now, mycorrhizae is a special kind of fungal mass that's living in association with plants. So that's a symbiotic relationship, helps plants grow, helps the plants absorb water. Okay, so the mycorrhizae is kind of a specialized kind of mycelium. All right, also had a question in the chapter 6 homework, which probably most of you haven't done yet because I haven't covered all of that yet. There's a question where it gives you 3 choices and then you actually have to type in the answer. So it's sort of an essay question. I have to go back in and grade that by hand because you type it out. So it won't tell you whether you're right or not, probably. So again, it's just one question, but it is a little bit confusing. I should probably just get rid of that one. So, all right. Anything else before we get started today? I think we'll get through the rest of chapter 6 here pretty quickly, and then we will start metabolism, but that won't be on the exam. All right, so we'll make a break there when we get to that point. All right, so after we got through the fungi last time, we started. Oh yeah, question. So is the test only multiple choice, or is there essay questions as well? Okay, good question. The test is only multiple choice. Yep, 56 multiple choice. Scantron. Okay, so we started talking about different environment. Oh, another question. I would say a pencil is your best bet, a dark pencil. I haven't found that the Scantron has too much trouble with, if it's a light pencil. I see students do it in pen. I don't think that's really not a good idea in case you want to change your mind. But yeah, bring a couple pencils, darker the better. Yep. Okay. All right, so we talked about environmental conditions and kind of what organisms need to grow. So we're going to talk now about specifically what does that growth look like? How do we describe the growth? How do we actually count cells in the lab? So to kind of finish up here. Okay, so bacteria grow by asexual reproduction. We call that binary fission. Okay, where essentially one cell gets larger and larger, almost doubles in size, and then splits in two. Okay, so there are some types of binary fission where they might not be equal, but for the most part, one cell and then it's splitting into two. Okay, eukaryotic cells are going to reproduce sexually by mitosis and cytokinesis. All right, and we're not going to worry about that. Okay, so binary fission, again, that's considered asexual reproduction. Okay, so you can see in this is a really nice a transmission electron micrograph there. So that's what a cross section of a cell might look like. You can see how this cell, well, it's hard to see this, but it's actually getting longer and longer. And if you've been looking at cells, if you're in lab this week, and you might have noticed that some cells are longer than other cells. Well, some cells are maybe just getting ready to divide. So they're going to be longer. A shorter cell would be a younger cell. Okay, but they're getting longer, and then you get this septum forming right in the center, and then eventually one cell becomes two. The time it takes for one cell to become two, we refer to that as the generation time. The time it takes for one cell to become two. All right, so we also refer to that as doubling time. Okay, well, should be like that. Okay. All right, so I like this little graphic here over the side here in your book. It just shows you've got one bacterial cell, it's getting longer, chromosomes are replicating, you get that septum form in between, and then basically, eventually that one cell becomes two separate cells. They may or may not stay stuck together. Remember, that's what we see different cell arrangements, whether they stay stuck together or not. But for this organism, 30 minutes. See, that varies tremendously depending on what species it is. But let's say 30 minutes. Okay, so that's the time it takes for one generation. And then, so those two cells are going to elongate and eventually make 4 cells. Okay, there's your second generation. That took another 30 minutes. And then 4 cells become 8. Actually, your book drew 9. I don't know why. That doesn't make sense. 4 cells become 8. That's the third generation, so on and so forth. 8 becomes 16. Okay, so that's typically the way bacteria grow. So yes, one cell is growing, but when we're talking about bacterial growth, typically mean changes in the population numbers. So, okay. So when they are doubling in number every generation, that's a kind of growth that we describe as exponential growth, or you can also call that logarithmic growth. And that's typically the way populations are going to grow unless there's something that's severely limiting their population. So if we graph logarithmic growth, it's going to look like the one on the right there. Okay. So basically, again, every generation, they're doubling in number. And you can see how the numbers get large very quickly. If you compare that logarithmic growth to growth that's not logarithmic, that would be more arithmetic, that just means that every generation, they're adding a set number. like, okay, every hour or whatever, they're adding 2 organisms. The next hour, they add 2 more. The next hour, they add 2 more. Okay, that population is going to grow, but very slowly. Again, this is only going to be if there's really something that's limiting some nutrient maybe, or, you know, the wolves on Isle Royale, like they're limited by the number of moose that are there, something like that. Okay, so the generation time, what kind of things is that dependent on? I spoke about that a minute ago. What species is it? Some organisms just have a much quicker generation time. E. coli under ideal conditions will have a generation time of about 20 minutes. Okay, so this is something that I had to do when I was in college in an upper level micro course. We had slides that had agar on them, and we put bacteria on there, and then we had to look at them under the microscope and just watch them grow. Okay, it was kind of boring, but you actually eventually did see a cell divide, and we could witness one generation. Some bacteria, their generation time is more like 24 hours rather than 20 minutes. So then in order to see visible colonies, it takes weeks and weeks and weeks of growth. So again, there's a large variation there. So what species is it? And as well as environmental conditions, like the things that we talked about last time, what temperature. We saw that E. coli grows a lot faster when it's at body temperature than what it does at room temperature. Are there nutrients available? Things like, you know, is the pH ideal? Is the oxygen concentration ideal? So all of those things. Other physical parameters. All right, so just another word on graphing out numbers of organisms. And of course, you have to have a way to count them, which we're going to talk about in a minute. But if we were to try to make a growth curve, so if we're going to do that on kind of regular graph paper, you can see here, so, you know, at each time we've got a count here. You can see that if you wanted to extrapolate from a growth curve how many cells you would have, say, at 10 hours, you're going to have some really long graph paper, right? Because it's going to be, you know, way up here somewhere. So what's usually more helpful is plotting your numbers on some semi-logarithmic paper. So if you have the time on the x-axis, and then if the y-axis is on a log scale, then it's going to give you actually a straight line. All right, so logarithmic scale, so you've got 10 here, maybe 100 here, 1,000 here, 10,000, that's a logarithmic scale. Okay, so you could plot if you plot your numbers, and if you have exponential growth, then that's going to give you a straight line. And you could say, well, let's see, how many cells am I going to have at 10 hours? You could, you know, run a plot over and then, you know, estimate how many cells you're going to have then. So that actually is a lot more helpful. Okay, so we could use a graph to kind of calculate or estimate your cell numbers. We're going to talk a little bit about how to do that with a formula, although I don't really want to get into having everybody need to bring calculators to class. So we're going to think through how to do this without calculators and just use sort of really manageable numbers. But it's kind of helpful to understand the formula because, you know, normally you are going to be working with pretty big numbers. Okay, so first thing, if we're trying to estimate the number of cells arising from a single cell, okay, we describe that as 2 to the n when n equals the number of generations. Okay, so if we have a single cell and we want to know after one generation, how many cells we're going to have? We're going to have 2. So one cell doubles and becomes 2 after one generation. Okay, if we want to know how many cells we're going to have after 2 generations, okay, that's going to be 4. 2 times 2. All right, 2 to 3. All right, 2 times 2 times 2, that's 8. Okay, so if we put that into formula, our number of cells, final count, is going to equal to how many we start with, the initial number, times 2 to the n. All right, so if we try just plugging in some, low numbers, right? Say we start with 1 cell, and we want to know after 1 generation, right? That gives us 2, right? Easy enough. If we start with, say, 100 cells, let's try this again, and we want to know how many cells we're going to have after, say, 2 generations. Okay, that's going to equal 100 times 4. Our final count is going to be 400 cells. Okay, so again, when you're working with small numbers, hopefully that kind of makes sense. We can kind of cast that formula off to the side though, and just think through this when we have larger numbers. So this is more like, I think there's at least one question in your homework that's kind of similar to this. Okay, so let's think through this. So assuming that you are working in a lab and your cells are growing exponentially. Okay, as we'll see, that's not always the case, but let's pretend that they are, just for sake of making a calculation. Okay, if you have 20 cells to begin with in a broth culture, and you know your generation time is 30 minutes, how long will it take until you have at least 1,000 cells? Okay, so it's given that we'll say your time 0, you have 20 cells. Okay, so if we think through this, that number is going to double every generation. Okay, so after 1 generation, you're going to have 40 cells. Okay, and we were told that that takes 30 minutes. Okay, so that's 30 minutes. All right, after 2 generations, you're going to have 80. That's going to be an hour. All right, 3 generations, double this again, 160. 1 hour, 30 minutes. 4 generations, 320. That's 2 hours. 640, 2.5 hours. 6 generations, that's going to be 1,280 cells. All right, so we wanted to know how long will it take until there are 1,000 cells. That's 6 generations or 3 hours, right? So you can kind of just think through that by doubling your numbers every time. The one mistake that people tend to make is that they say they count 20 cells as their first generation, and then they go on to their second generation. So that's 20 is what you start with, times 0, and then 1 generation is double that. Okay. All right, so that's a little bit about numbers there. Before we get into counting cells, we need to understand, like I alluded to, that it's actually not true that all cells are always going to be growing exponentially as fast as they can. There are actually phases of microbial growth. Again, we're talking about in a closed system like a test tube, okay, when we can count them easily. Typically, so here's your time 0 when you inoculate your test tube. Okay, are they all of a sudden growing exponentially? No, we almost always see that it takes those cells a little while to acclimate to their medium, maybe start making the correct enzymes in order to metabolize the nutrients that they're given. You almost always have some kind of a lag phase where the cells are either growing very slowly or not at all. Okay, let's see, or Okay, and then once they acclimate to their medium, they're going to grow exponentially and enter into a log phase. This is so they're growing at their fastest rate. The cells are very uniform in size. This is actually the phase of growth where the cells are going to be most susceptible to some kind of antibiotic. Remember when we talked about a growing cell, most antibiotics are going to work best when the cell is growing. We talked about those autolysins that cells make to cut the bonds in the sugars of their cell wall in order that new subunits can be put in there, and then transpeptidases have to kind of reseal those amino acid linkages. Okay, so that's the time when that cell wall is most kind of susceptible. And so when antibiotics act there on the growing cell, then that's when they're most likely to work and cause the cell to die. So, okay, but they can't keep up that exponential growth forever. Eventually, they're going to run out of nutrients. The waste products are going to build up. Waste products are often quite acidic. Okay, so at some point, you get your growth is going to be equal to your death of cells. Okay, and that growth levels off and then eventually will decline. Okay, where you have more cell death than you have growth. It takes a while for every last cell in that culture to die. Like really, you know, lots of waste products growing up, nutrients depleted, but eventually that will happen. Okay. All right, well, sometimes we want to know what stage our culture is in, so it's advantageous to know whether they're growing or not. So I want to investigate some different ways to measure bacterial growth. 2 ways that require incubation, and then we'll talk about 4 different ways that don't require incubation. Okay, so the 2 methods that require incubation are the viable plate count, which should be familiar if you're in lab. We're going to talk about it briefly here, though, as well as membrane filtration. All right, so I'll come back to this slide in just a minute. This is your textbook schematic of what you're doing when you're doing the viable plate count. Okay, so essentially, they don't really show the original culture here, but we're trying to figure out how many cells are in that original culture. And so you're just basically going to make a series of dilutions and then put an aliquot of that dilution onto an agar plate. When we did this in lab, we spread that around with a hot glass hockey stick. And then we incubated and see how many colonies grew up. So the idea is that each cell in this original culture is going to grow into a colony on the plate. Okay, so we can extrapolate back to how many cells were in the original culture by how many colonies that we count. So that's why we have to make dilutions, though, because if we take it straight from the culture, you're going to usually have way too many to count. We were looking for kind of a sweet spot between 30 and 300 cells so we can count it accurately. Okay, so here we would do a calculation with this plate. These would not be considered statistically significant. Okay, so you take into account the dilution here and how much you put on your plate to figure out how much was in your original sample. Okay, we're not going to do that calculation in here. I'll let your lab professor worry about that, but that's just kind of the background, the viable plate count. The other method that requires incubation is membrane filtration. So you're going to use this method if you have some sample that you think actually may need to be concentrated in order to give you a statistically significant number of cells to count. So when would that happen in nature? Almost never. I've done this with water from the Grand River, and I thought I would have to concentrate it, and actually I had to dilute it to get it to be in a countable range because there was a lot of stuff growing in the Grand River. So really the only time that you would want to do this if you're trying to test some treated water, so tap water. Okay, so you get, you know, a known quantity. So say 100 mils. Whoops. Okay, sorry about that. You get maybe, you know, whatever, 100 mils, and you pour it into your contraption here that's got a membrane right here. And then you're gonna have to use a vacuum to suck that water through the membrane because the pores are really small. And then you take off that membrane and put it on a nutrient medium and agar medium. You let it incubate and grow up. The nutrients will kind of come up through the membrane. And once again, you count the number of colonies, and having that known volume up here to begin with, you can figure out how many cells you have per mil. So we often do this, again, like I said, if we're testing tap water, usually we're looking for contaminants that come from some kind of fecal contamination. So we have a special media where those bacteria that we don't want to see are going to be green if they grow up. So kind of a differential media. So some colonies look different than others, and we know something about those colonies based on the color change that we get. Okay. So the nice thing about those two methods are they both only count live cells. Okay, viable plate count makes sense. Usually it's the live cells are the ones that we're interested in. Okay, a drawback though is that they take longer. Okay, you've got at least an overnight incubation depending on how long it takes the colonies to grow up. So you don't get your counts back till at least another day. Sometimes that's fine. Sometimes you want your numbers longer than that. But they're both cheap as well. No fancy equipment needed for either one of them. Okay, so now we look at some methods that don't require incubation. The first thing we think of is using a Petroff-Hauser chamber. Another name for that is a hemocytometer. Might be a little bit easier to remember. But this is just a modified microscope slide where there's actually a grid etched here and here on that microscope slide. You can't see it unless you look through the microscope. And then we typically will put a cover slip on here like this. And then you can kind of see it right here. You pipette in your solution, your microbial solution that you want to count. It kind of goes into that little ridge right there. It wicks underneath the cover slip uniform width. And then you put that under the microscope and focus on the grid and count a certain number of squares. And you can extrapolate back from that numbers that you get in a certain number of squares to the volume of your, original sample. So concentration in your original sample. Okay, so you would probably want to do that, a number of times to get kind of a good sample size to get the most accurate number. Again, it's pretty quick method as long as you can see your cells well and know how to do the calculation and all that. It's a good method. Another method is using a spectrophotometer. Okay, so remember how spectrophotometers work. You usually, whatever medium you're using, you have to zero it out. So for here, we might take some sterile media. So zero the spectrophotometer out, and then you shine light through there at a particular wavelength. And what you're testing is the ability of that liquid to absorb light. So if you had nothing growing in here, the light would go right through. But the cloudier your culture is, then the more it's going to, it's more it's going to absorb that light. Okay, so the higher absorbance readings are just measuring its turbidity. Okay, so actually able to measure kind of the cloudiness of your sample. Okay, also called turbidity readings. And we know the cloudier your sample is, the more cells are there. Okay, so this is really good if you want to see if your culture is actively growing. Take out an aliquot every hour. Oh, is the absorbance higher? Okay, yep, it's growing. It doesn't give you actual numbers unless you make some kind of a standard curve. So that maybe, you know, the first time you're working with a particular species in a particular medium, you know, you would take absorbance readings, and then you would have to also do counts, maybe some other method, like with a Petrov-Hauser chamber. And then you could make a standard curve like this. And then the next time you knew when you got a certain absorbance, you would know, oh yeah, that correlates to whatever numbers. So, okay, quick, just like the Petrov-Hauser chamber though, it's kind of hard to tell if you have dead or alive cells. Well, impossible with a spectrophotometer. Difficult to look at cells under the microscope and tell if they're dead or alive. Impossible here because dead cells are going to scatter light the same way as live ones. Okay, so now we're kind of going up a little more money here, little fancier equipment. A coulter counter is also called an electronic counter, we'll just say. Okay, so these are going to get into probably thousands of dollars. The premise here, you put your bacterial solution in some kind of container, and then the counter will pass that solution through an electric field. Okay, so each cell will disrupt the electrical field enough that it will then keep track of that, and it will give you your counts. So really fast. It does work better for a dilute sample. Oops, 1L. Okay, so you may need to dilute your sample a little bit and then make some kind of calculation. And fortunately, bacteria can tend to clump together a little bit, so that can throw off your accuracy. So it's not good for cells that clump or cluster together. Okay, and again, you can't tell if it's dead or alive. Dead and alive counted the same. Okay, so there are some drawbacks there, but certainly it's a lot easier to do than some of our earlier methods. There's one in the upper level microbiome lab. I haven't used it, but we do have one. Okay, and then kind of the best of the best. Okay, lots and lots of money here for a flow cytometer. One that has all the bells and whistles to it can run up to like $1 million. We do have one at Grand Valley that's a much pared down version of that I believe was much less. But these are pretty fancy pieces of equipment. Basically, they not only count cells, but they can separate out cells. If you have, you know, say 3 or 4 different kinds of cells in a population, it will count them and separate them out at the end, which can be useful. Okay, I know we had one of these when I went to college, where I worked in a lab for a while, and the university employed a PhD scientist that it was his job to work the flow cytometer and help any of the researchers that wanted to use it. Like that was all he did. So kind of like electron microscopy. Start video. Okay. Well. All right. Okay. Still recording. All right. Good. So there's 3 different colors here, 3 different fluorescent dyes. So you have 3 different, different kinds of cells here that you can kind of visualize there. So anyway, do you need that level of specificity? Maybe oftentimes not, but it's available. Okay, so that finishes up chapter 6. And that's the end of the material that I want to test you over for Tuesday. Okay, any questions with that you can think of at this point? Okay, we'll press on. Metabolism is usually not students' thing that they really look forward to studying. It is maybe a little bit drier, but I hope that you can kind of appreciate it a little bit. We are not going to be studying metabolism to the level that maybe certainly you will in your biochemistry classes. We are not going to be memorizing pathways. So I'll try to give you a good idea of kind of the level that I want you to know things, both in class and when you take a look at the learning objectives. This is chapter 5. Hopefully that will I'll make that as clear as I can. But don't be afraid of it. Again, we're going to take more of an overview approach. And we're specifically interested in bacterial metabolism. So we're going to be looking at some things that bacteria can do and that we as humans can't. Okay, so we're going to do a little bit of a review of the components of metabolism, some of the terminology that we use, just so we're all on the same page there. We're going to work our way through aerobic respiration, so glycolysis, Krebs cycle, electron transport chain, and then spend some time on fermentation and anaerobic respiration, which again are types of metabolism that are very common for bacteria. All right, so first terms that hopefully you recognize the difference between an anabolic reaction and a catabolic reaction. Okay, an anabolic reaction, that's a reaction that builds molecules. In other words, a biosynthetic reaction, biosynthesis. Okay, that's always going to require energy. Another way we say that, it's an endergonic reaction. Okay, and then a catabolic reaction, these are the ones that we are primarily interested in because they are the ones that are giving us energy. Those are catabolic reactions. Okay, so reactions that break down molecules. Okay, and of course, the anabolic reactions and the catabolic reactions in the cell have to be pretty tightly balanced. Okay, you don't want to make too much energy that you're not going to use, and of course, you don't want to be making structures that you don't have energy to build. So, really well balanced. It's a nice little graphic in your book here. A bacterial cell here, it's absorbing some nutrients. So a lot of times they will secrete some kind of enzyme that will break down a larger molecule, maybe outside of the cell, so they can bring the building blocks or the nutrients in. Remember, they can't phagocytize because they have a cell wall. And then in some catabolic reaction, okay, they're broken down further into precursor molecules, we might call them. It's not a 100% efficient reaction. You always lose a little energy as heat there. And then in anabolic reactions, those precursor molecules are used to build macromolecules, proteins, lipids, nucleic acids, carbohydrates. Okay, and those are used to build cellular structures like membranes or whatever, ribosomes, whatever, to grow the cell, or maybe for storage as well. Okay, and again, always going to lose a little bit of energy as heat. Okay, just like your car engine. Redox reactions. Okay, inherently confusing. I think this YouTube video is really kind of fun and actually kind of helpful, especially the first few minutes of it. It's from Crash Course Chemistry, and the guy kind of talks through redox reactions a little bit. One thing that he is fond of saying is that it's all about the electrons. Okay, so we are particularly interested in where the electrons go because they are what gets converted into the lion's share of ATP during respiration. Okay, so redox reactions, that we have to kind of understand where the electrons are going here. Most of you are familiar with the mnemonic oil rig. Okay, oxidation is loss of electrons. Reduction is gain. Remember the of electrons part. Okay, so here if you have an electron donor, okay, that cell becomes oxidized when it loses an electron. Okay, now it can gain an oxygen, but you know, sometimes it happens, sometimes not. Okay, here, so the electron acceptor, it becomes reduced when it gains the electron. So why do we say when it gains something, it's getting reduced? He admits this is kind of a poor way that we have named it, but kind of you stick with, you get a name and then it's hard to change sometimes. You could think about the net negative charge is reduced. You could kind of think of it that way when a molecule is gaining an electron. Okay, I think one of the more confusing things here with redox reactions is that we forget that protons very often accompany, well, let me try that again. Can't spell accompany. Protons very often accompany electrons. Okay, so we don't often draw the electrons in a chemical formula, but we almost always put the protons in there. So when a compound is gaining a proton, you assume it's gaining an electron as well. Okay, so again, that can be definitely a little bit confusing. Remember, a hydrogen atom is equal to a proton and an electron. Okay, so again, usually that proton and electron are traveling together. All right, we don't let electrons just go wherever they want in the cell. It's kind of like some teenagers at a dance, right? We always chaperone them because they could get into trouble on their own, right? Electrons are the same way. They can cause a lot of damage if they get loose in the cell where they're not supposed to be. So we have these electron carriers that escort the electrons to where they're supposed to go. NAD, so this is the oxidized form of the carrier, nicotinamide adenine dinucleotide. NAD can actually hold a hydride ion. And a hydride ion is equal to 2 protons and an electron. Okay, so the reduced version of NAD is NADH plus H plus. Sorry, I think I said that wrong. Sorry, other way around. It's 2 electrons and 1 proton. Okay, so there's actually right here with the NADH, okay, it's carrying 2 electrons and a proton, and then there's an extra proton left over. Okay, where does it go? It doesn't matter. They just can go free in the cell and they don't cause any damage. FAD can actually carry 2 full protons. So when FAD is reduced, all right, that over here, reduced, it's FADH2. And then there's another carrier, NADP, that also carries a hydride ion. So it's going to be NADPH plus H plus. All right, so again, there's two electrons riding on there with that proton. Okay. All right, what else do we need to review? Of course, ATP, the energy currency of the cell, adenosine triphosphate. Of course, this is recyclable between ADP and ATP. So the energy here in ATP is carried in these high energy bonds between the phosphates. And when one of those phosphates is cleaved off, that gives the cell energy to do some kind of work. Okay, so it cycles between ATP and ADP. Okay, and when the cell gets energy either from sunlight, if it's a photosynthetic cell, or from ingesting some kind of nutrients, then it gives the cell the ability to add a phosphate back on to ADP to make ATP again. Okay, so there's actually 3 different ways that we describe that ADP gets phosphorylated back to ATP. Okay, so I want to go through those 3 ways. There's really only 2 that we're going to be concerned with. The first way is by substrate level phosphorylation. Okay, and I've just put glycolysis up here. Just briefly, we're going to get to it in more detail in a little bit. But during glycolysis, this is an example of when ADP gets phosphorylated by substrate level phosphorylation. And that name's a little bit confusing. It generally means that the phosphate comes from some other substrate in the reaction. That's not always true. Sometimes it's kind of floating around in the solution. But in this case, here we have a molecule in glycolysis that has 2 phosphates on it, 1,3-bisphosphate or diphosphate is the way it's usually said now. So one of those phosphates is ripped off of that molecule to phosphorylate ADP to ATP. Again, we call that a substrate level phosphorylation, and that happens again. Then the other phosphate here is stripped off again. So we end up getting 2 ATPs out of kind of the bottom half of glycolysis, and both of that is during substrate level phosphorylation. The other way that ADP gets phosphorylated back to ATP is called oxidative phosphorylation. Now, this doesn't necessarily mean that oxygen is doing this. What actually this means is that NADH is getting oxidated back to NAD. Okay, so this is happening at the electron transport chain. And we'll talk more about this soon, next week. But this is kind of, the electrons get dropped off at these electron carriers in the cell membrane. So I like this kind of analogy here of the dam. If the dam here is the cell membrane, right, and the electrons that NADH is carrying, they get dropped off at the cell membrane. Say this is a cell membrane. Okay, and as those electrons get passed down here to some acceptor at the end, it's pumping protons out here. So where the water is, okay, all those protons that get pumped out while the electrons get passed from the carriers that's representing lots of potential energy, just like in a dam. And when those protons are allowed to flow back through, say at one spot, then that powers the phosphorylation of ADP back to ATP. Okay, so that's why when you say it's all about the electrons, it's because it's the electrons that get converted into kind of the lion's share of ATP that we get from respiration. Okay, so oxidative phosphorylation is always going to occur at electron transport chains. And it could be that oxygen is the final electron acceptor, but it could be something else. Bacteria are able to use other things as electron acceptors, usually kind of some kind of inorganic ion. That's where oxidative phosphorylation takes place. The third type of phosphorylation is photophosphorylation. Okay, that's going to happen during photosynthesis. We're not going to go into photosynthesis in this course, so I'll just mention it here. But again, there's no disease-causing organisms that photosynthesize, so we're not going to worry about that. Okay. All right, and let's lay some groundwork talking about enzymes, or reviewing enzymes a little bit. Hopefully it's review. Okay, so of course, what do enzymes do? They speed up reactions. Okay, so reactions that are favorable, that would take place anyway, but that would take them so long to happen that life wouldn't be supported. All right, so another way to say that is that it lowers the activation energy. It makes the reaction a lot more likely to happen. The terminology that I typically use is enzymes act on substrates. Oops. Or here they call it substrates reactants. Okay, and what you get is some product that's different from the substrate. Okay, so the substrate has been changed, the enzyme is not changed. Once it catalyzes that reaction, it goes and changes other substrates. Okay, if you ever see something that ends in ASE, like lipase or catalase or superoxide dismutase, you know it's an enzyme. There are some enzymes that don't end in ASE, like trypsin, for example, an enzyme in your stomach. Okay, so that's a little bit annoying, but ASE is an enzyme. If something ends in OSE, then that's a sugar, like glucose. Okay, so don't confuse those. Most enzymes are proteins. Okay, but of course they can actually require some kind of non-protein component in order for the enzyme to function. Okay, so if there is a non-protein component that is required, we call that a cofactor. If it's some kind of inorganic requirement. So here you can see an inorganic cofactor, something like, say, copper, for example, or iron might be necessary. We call it a cofactor. If it's an organic cofactor, then the correct term is coenzyme. Okay, so in this picture here, this enzyme, the protein component is referred to as the apoenzyme. And then because of the coenzyme and the cofactor, those both play a role into the shape of the active site. And of course, the active site is where the substrate binds. So if it's not just a simple enzyme is just protein. I'll make a note of that. Simple enzyme. Then the terms that includes the the cofactors or the coenzymes is a holoenzyme. Okay, so the apoenzyme is the protein part. The holoenzyme is just the whole thing. And organisms, to some extent, oops, oh, I forgot. This, I don't know if I posted this or not. This is just an example of an enzyme. It's an enzyme that works during glycolysis. Don't memorize this. You don't need to know these structures or what the enzyme is. But, you know, here's a very important enzyme in glycolysis. It's one that actually splits the sugar into two. The substrate binds here to the active site, and then that substrate is changed. It's actually split in half. Okay, and then the enzyme goes on to work on some more substrate. So just some example of enzyme function. Things that influence enzyme activity, same kinds of things that influence the growth rates of bacterial cells, things like temperature. There is an ideal temperature for enzymes. It's, you know, different for different cells, where the enzyme activity is the fastest. But at some point you get a little higher than that and the enzymes can't function anymore. They become denatured, oftentimes permanently, so they're not going to work. Of course, there's going to be some ideal pH. Usually there's a little bit of range that the enzymes will function, but there's going to be some ideal. And then if you think of things like substrate concentration, to a certain extent, okay, the more substrate that's around, the faster the enzyme can work. But at some point, there's a saturation point, because if you have more substrate than what you have enzyme, then the enzyme can't work any faster. Okay. Usually cells, to some extent, want to control the activity of their enzymes in order to control their metabolic reactions. We will see a few weeks from now, when we get into our genetics, how a cell can actually control whether an enzyme gets made or not. So it can save energy that way. But once the enzymes are made, it may be to the benefit of the cell to kind of shut it down for a while. All right, so there's different kinds of inhibition. A competitive inhibitor is some compound that's going to block the active site. Okay, so remember, the active site is where the enzyme binds. So a competitive inhibitor is going to kind of have the same shape as a substrate, most likely. All right, so you can see that it's bound here instead of the substrate, and so there's no product formed because the substrate couldn't bind. Okay, but this can be overcome to some extent if there's more substrates than inhibitor. Okay, so that's one way that can be overcome a little bit. Another kind of, I decided to not call this inhibition exactly, and I'll show you why in a second. But if it's some kind of non-competitive control, that means that well, I'll go ahead and call it an inhibitor, binds to some other site, not the active site. Okay, so I'll put other in parentheses. We have a term for other, we say allosteric. Okay, so an allosteric site is one that's not the active site. So over here, active site is up here. Here's some allosteric site where the inhibitor binds. And when that inhibitor is bound, that actually affects the active site, though. Changes the shape of it a little bit so that the substrate can't bind anymore. Okay, this can actually work not as an inhibitor, but as an activator. So there may be an instance where something has to bind at an allosteric site in order to change the shape of the active site, so that now the enzyme substrate complex fits together and becomes functional. So only then can the substrate bind. Okay, this allosteric control, another example of that is during something called feedback inhibition. So typically this is where you have a pathway that involves, several different enzymes in order to get some end product. What if that end product starts building up? It'd be helpful if there was some way to kind of shut that pathway down so the cell stops kind of wasting energy. So here you start with a substrate, and again, over a series of reactions, you get come an end product. If there's too much of that end product can perhaps bind allosterically to that first enzyme in the pathway, shutting it down so that that original substrate no longer fits into that enzyme. Okay, this is always, it's hard to say always, right? But usually going to be some kind of allosteric inhibition. If it was competitive inhibition, then that end product would be the same shape as that original substrate. So that wouldn't typically be the case. So it's usually going to bind at some other site other than the active site. All right, so that's kind of our little review of terms and kind of the groundwork of metabolism as we start to go through pathways. So keep in mind as we move forward and start to look at pathways, we're going to pay particular attention to where the cell is gaining ATP. Okay, how much does it start with? What is it? How much ATP does it get out of it? There are key juncture points. So we are not, like I said, memorizing pathways or enzymes or structures. We're going to pay attention to what you start with, what you end with. If there's kind of a big decision point where sometimes the cell will use this pathway or sometimes the cell will use that pathway, we're going to pay attention to that. And what I mean here is, when would a cell use this pathway? When would a cell use another pathway? Does it ever use both of them? So kind of big picture things. Sometimes it's helpful to count the carbons. Okay, if you're starting with a 6-carbon compound, you're going to end with 2 3-carbon compounds. And if you don't, well, what happens to that carbon? Okay, so we are going to pay attention to that because that's not too hard to keep track of. And like I said, we're going to pay particular attention to the electrons and where they go and how they get converted into energy. Okay, so I like this overview figure that your book has. I'm going to go back to this several times. It's kind of a nice, just map of what's going on here. Of course, in green here, that's respiration. All right, so we think about respiration as breathing in oxygen and respiring CO2. That's kind of macroscopic respiration. On the microscopic level, we think about it a little bit differently. I lost my train of thought. We think about it a little bit differently. So we think about, well, what's the electron acceptor? What's starting it? It's a catabolic reaction. I lost my train of thought there. Sorry, I'll get back to that. But you'll see here that glucose glycolysis is actually part of respiration pathways as well as fermentative pathways. Okay, sometimes that can be a little bit confusing, but a respiration pathway is always going to involve some kind of final electron acceptor. Like I said, that can be oxygen, but for bacteria, it could be something else, like maybe nitrates or sulfates, some inorganic ion. Okay, and the pathway looks similar depending on whether it's oxygen or some inorganic ion. Okay, so both of those are respiration pathways. And then fermentation involves glycolysis, and then, maybe just a few other reactions, something that doesn't require any oxygen. Okay, so we're going to start out with glycolysis and move our way through respiration first. and then talk a little bit more about anaerobic respiration and fermentation. Okay, so again, paying particular attention to where the electrons are coming off, where ATP is gained. All right, so I have a couple of different graphics of glycolysis. I show you a couple different ones because some of them are better at showing certain things and others are better at certain things. This is a really good name for what's going on here. The splitting of sugars. So glyco, we think sugars, lysis, splitting. Okay, so we usually start with glucose. Why do I start with glucose? It's usually the most readily available sugar. If you start with some, say, disaccharide, typically that disaccharide is split into its monosaccharides, and then they funnel into glycolysis right to begin with. Glycolysis is something that is almost every single organism does it. The exceptions are a few bacteria, and we're going to talk about that pathway that's just a little bit different than glycolysis. But like I said, everybody's doing it. Like almost all of life does it. We do have another name for it, which can be confusing when these pathways go by different names, but it's also called the Embden-Meyerhof pathway. Sometimes Embden-Meyerhof harness. Okay, we like to give homage to the people that elucidated the pathways. So that's why sometimes you hear this referred to as Embden-Meyerhof, or sometimes just the EMP pathway. Okay, and you kind of just got to recognize those alternative names. Sometimes in papers, I see them talking about Embden-Meyerhof, and you just got to know that they mean glycolysis when they talk about that. Okay, so almost all of life does it. Again, just kind of a little overview of what's going on. Energy is invested to begin with. So we've got 2 ATPs being invested. We have this splitting step. Now this fructose 1,6-bisphosphate is not a symmetrical molecule, so you can't just split it in half. This is a little bit of a simplification here, but the next picture shows that a little bit better. And then down at the bottom half here is where you get the energy generated. So you get the electrons coming off of here and harvesting of ATP. So what do we end up with from one glucose? We end up with, we put 2 ATP in, but get 4 out. So we net 2 ATP, we net 2 NADH. Okay, those are gonna drop off their electrons at the electron transport chain if the organism is able to respire aerobically. And we end up with 2 pyruvates. Okay, or pyruvic acid, same thing. Okay, so organisms that actually can't do respiration and can only do fermentation, sometimes this ATP is the only ATP, that's the only way that they're able to generate ATP. Okay, so it doesn't seem like a lot, but it's enough to keep them going. Okay, this is the graphic of glycolysis that your book shows. This one's kind of nice because it actually draws in the carbons. So you can kind of keep an idea of, well, I'm starting with a 6-carbon compound, and again, I end up with 2 3-carbon compounds. If you happen to have an old edition of your book, the old graphic here used to just say, after the splitting step, it would say, oh, we have 2 of this, 2 of this, and 2 of this. And they decided to make that more obvious where the split was and split it up. And in the old edition, they kept, oh, we got 2 of this and 2 of this on one side and 2 of this and 2 of this on the other side. So it had you ending up with 4 pyruvic acids instead of 2. Again, the new edition, they fixed that. So hopefully that won't mess you up. But this does show that, you know, when you split here, you're actually not getting 2 glyceraldehyde 3-phosphates. you're getting this dihydroxyacetone phosphate, but then that usually gets converted into glyceraldehyde 3-phosphate. So just a note that they're sometimes that extra step they don't really show. It's nice that they show the phosphates here. So again, it's ripping a phosphate off of that molecule to phosphorylate ADP to ATP. And remember, we call that phosphorylation by substrate level phosphorylation. Okay. So, you know, really that's as detailed as I want to get into with glycolysis. You know, I mentioned some intermediates there, but that's not anything that you need to memorize. We're not worried about the enzymes. Of course, it is enzymes. It's facilitating every step. And when you do actually look at the structures and you see what the enzyme does, then I think you really appreciate it. So don't dread doing that in biochemistry. I think that it definitely gives you a good appreciation of what's going on. So I mentioned that there are some bacteria that are not able to do glycolysis. It's because those bacteria, I think it's this enzyme that works right here that some bacteria are missing. So they actually do an alternative pathway to glycolysis. It's called the Entner-Doderoff pathway. And that's a little bit confusing because it sounds like Embden-Meyerhoff. But that's, again, this is a separate pathway. Oops, sorry, lost my pen. Oh, there we go. Only some bacteria are able to do this Entner-Doderoff. Okay, we don't do it. We stick with glycolysis. Okay, but I like how this graphic compares the Entner-Doderoff to the Embden-Meyerhoff. And your book really only gives a paragraph or so to Entner-Doderoff. So we won't spend a lot of time on it here. I think the pathway might be in the back in an appendice, but maybe not. The differences here, and I know we're running out of time, but first of all, because it doesn't have this enzyme, it starts off a little bit different right from the get-go. We're using a different electron carrier here. So the difference is because it's that phosphate actually tags those electrons to be used for some biosynthetic reaction. Those electrons are not taken to the electron transport chain. Okay, so that's one difference. And in the splitting step here, we right away get a pyruvate coming off. Okay, so the cell kind of loses the ability to gain all the stuff that normally is gained in glycolysis in the bottom half of the pathway there. Okay, but then this part is just the same as what we see with glycolysis. So differences here are at the top. Net gains for Entner-Doderoff, only 1 ATP. Okay, so we input 1 and get out 2. So 1 net gain, we get 1 NADPH, some electrons, and we still get 2 pyruvate. Okay, so similar, but only half the amount of energy. Is that a problem? Usually organisms that use this pathway are able to respire aerobically, so they're getting a lot more energy elsewhere, so that's typically not a problem. What kind of organisms do that? There's just a few examples I put here at the bottom. E. coli actually only does it when we grow it on certain medium, but there are other organisms that's what they do all the time instead of glycolysis. So we will pick up there in a week. Remember, I have office hours tomorrow if you want to talk to me. Have a good day, good weekend.